So I wanted to take a minute and go through two sort of slides that we have, or two homework questions that we have too, that are kind of dense and intense, but I want to make sure that I give you sort of something that you could rewatch, re-listen to sort of understand these two systems. We're going to be looking at kind of thinking about high blood sugar and low blood sugar. And we're going to take a first sort of a deep dive as to thinking about the different enzymes that are responsible in regulating this sort of cascade. So that's kind of looking at this image. And then we've got kind of how this is controlled and how we think about it from a tissue standpoint in terms of muscle cells versus liver cells. So again, I'm going to start with this slide. This slide again highlights some different aspects of how we put ourselves into either a phospho or a diphospho state, specifically thinking about glycogen breakdown here. And so to kind of start out, and we're going to do a little bit of structural review here, have some details here that if you want, you can sort of go back and look at these structures. Remember, this is for glycogen phosphorylase. This is for pKa for the catalytic subunit. This is for the regulatory subunit of pKa. And then we sort of learned about phosphorylase kinase. So I'm going to be referring back to these, but I'm not going to flip back to these figures. But let's take a second here and map on what happens, kind of thinking about a high blood sugar and a low blood sugar situation here. Before I go on to the next slide where we kind of have some words with it, I wanted to use this slide because it kind of highlights it with the structures and kind of some information that we know. So let's first think about what happens under low blood sugar levels. Since we've kind of learned about glycogen breakdown first, that might make the most sense to sort of think about here because we've got this phosphorylation cascade to think about. So again, if we've got low blood sugar levels, that means your pancreas is secreting glucagon. And glucagon is an external signal that when it binds to cell surface receptors, it's going to activate adenylate cyclase. So activating adenylate cyclase is going to be what's responsible for making cyclic AMP. So remember that pKa is an enzyme that exists in an inactive tetramer where our regulatory subunits are bound up with our catalytic subunits. The regulatory subunits have that inhibitory domain that's bound in the active site of our catalytic subunit. However, as we increase the concentration of cyclic AMP, we can sort of think of distracting that regulatory subunit because it's going to bind to those cyclic AMP molecules. When it binds to those cyclic AMP molecules, conformation changes and it removes itself from the catalytic subunit. And in removing itself from the catalytic subunit, now that inhibitory domain is no longer bound in the catalytic subunit's active site. Again, we've got that active site with that threonine 197 that's already phosphorylated. So pKa is ready and available to do its phosphorylating. As you'll see here with these arrows, it actually phosphorylates multiple things. We're just going to kind of keep ourselves in this phosphorylation cascade right now. But we're again going to highlight that what it phosphorylates first downstream here in our cascade is the alpha and the beta subunits of phosphorylase kinase. So, highlighting some similarities with phosphorylase kinase to pKa. It is a tetramer. It's got 4 subunits here labeled with Greek letters alpha, beta, gamma, and delta. The alpha and the beta subunits are the regulatory subunits. So they're like the R subunits that we have with pKa. There's also this delta subunit, which is a calcium binding subunit. And then the gamma subunit is equivalent to the catalytic subunit. The gamma subunit is where we do our phosphorylating. So only one catalytic subunit that we have here in phosphorylase kinase. And one thing that's also different is when we make it active, it's not from a dissociation of subunits. We actually just have a conformational change of subunits. That conformational change allows for the gamma subunit to pull out its own inhibitory domain and then be able to bind and phosphorylate its substrates. So again, highlighting what's happening here, pKa's catalytic subunit is phosphorylating both the alpha and the beta regulatory subunits. Not explicitly drawn here, but calcium is going to be binding to the delta subunit. A large conformational change, not a dissociation, a conformational change of all of these subunits allows the gamma subunit to pull its inhibitory domain out of its active site, and now it can do its own phosphorylating. We're going to just kind of consider the next kind of player in line in this phosphorylation cascade for breaking down glycogen, which is to phosphorylate glycogen phosphorylase. Remember, this phosphorylation site is serine 14. So once we phosphorylated serine 14 on the N-terminus, we are not going to be burying that N-terminus. We're going to lock in that N-terminus on the outside by interaction with arginine residues, and now glycogen phosphorylase is ready to break down glycogen. So that's our phosphorylation cascade if we have a low blood sugar level. And again, we're going to map this on our next slide here. Let's kind of take the flip side of that, and let's talk about what happens if we're in a fed state, if we've got high blood sugar levels. Well, what really is going to be happening here is we're going to be talking about having phosphoprotein phosphatase 1, or what's referred to as PP1. So we've got PP1 here that is going to be active when we're in a D-phospho state. Okay, so we didn't talk a lot about, we had a slide on it, but we didn't spend too much time talking about this. We have phosphoprotein phosphatase 1 that it is in its most active state when it is actually phosphorylated by an insulin-stimulated protein kinase. And I know that was a really weird thing to think about, but when we've got PP1 that's activated by that insulin-stimulated protein kinase, it's actually going to be going around and snipping off phosphates. So what happens is if PP1 is active, it's going to be snipping off a lot of these phosphates. And again, what we can see here is snipping off these phosphates, put these guys in their D-phospho state. Their D-phospho state is inactive. If you've snipped a phosphate off of synthase, you actually make it active. So again, we've quieted the phosphorylation cascade, which means glycogen breakdown is not going to be happening, but we've activated the glycogen synthesis pathway by activating synthase. The last little piece here to kind of highlight is the fact that we also have sort of a fail-safe mechanism here. We've got a molecule here called phosphoprotein phosphatase inhibitor, or this is the PP1 inhibitor. And again, this PP1 inhibitor, if we are in a phospho state, is actually going to be activated, which means it's inhibiting PP1. So we didn't specifically talk about this yet here, but what we can see is if we activate the inhibitor by phosphorylating it, and pKa actually phosphorylates the inhibitor. If we phosphorylate the inhibitor, it's actually going to sequester PP1 and keep it quiet. So the logic for this makes sense, right? If pKa is doing all this work to decorate everybody with phosphates, we actually want to make sure that the guy who's responsible for undoing that decoration is inactivated. All right, so now let's map this on to a situation where we've got high and low blood sugar. And I know this slide is really dense and complicated, so let's just kind of break it apart into three sort of sections here first. So what we're going to be talking about is what happens with these either signals or enzymes. So that's kind of what's happening here. Then we're going to say what's happening with them under a low blood sugar state or a high blood sugar state. So just like we did on the last slide, let's first talk about what happens under a low blood sugar state. Okay, so again, low blood sugar state means we've got kind of a need state signal in your pancreas secreting glucagon. So your pancreas is going to secrete glucagon. That's going to activate adenylate cyclase. Adenylate cyclase is going to make cyclic AMP. Cyclic AMP is going to initiate this cascade because pKa's regulatory subunits are going to bind to cyclic AMP, dissociate from the catalytic subunit, and now pKa is active. Kind of shown on this last slide here with all of these arrows that are sort of coming out here. That's what I'm doing here and highlighting in green all of the downstream substrates that are sort of activated by pKa. So again, three of them we're going to have here. Let's talk about them sequentially here. First substrate kind of in our cascade that we activate is phosphorylase kinase. So phosphorylase kinase, remember, gets phosphorylated on the alpha and beta regulatory subunits, conformational changes in conjunction with binding calcium to calmodulin is going to allow that gamma subunit to remove its own inhibitory domain and become active. Once phosphorylase kinase is active, phosphorylase kinase is responsible for phosphorylating phosphorylase on that serine 14. Again, we talked about if that serine 14 is phosphorylated, it's not buried, it's locked in with those arginine residues on the surface. It's locked in with those arginine residues on the surface, and we now have an active phosphorylase. So now if phosphorylase is active, phosphorylase is going to be activating the breakdown of glycogen. So at the end of the day, breakdown of glycogen is responsible for remedying this low blood sugar state. And again, this is happening in the liver. So let's finish out our cascade here, right? Because if pKa is decorating things in this phosphorylation cascade to allow for the breakdown of glycogen, it's also phosphorylating synthase, which puts it in an inactive state. And we highlighted we don't want to be having phosphorylase being active and breaking down glycogen, and at the same time synthase making glycogen. That would be what we call a futile cycle. So that's not a good thing. So pKa actually makes sure that synthase is inactive. And the second thing that it also does is by phosphorylating the PP1 inhibitor and activating it, activating the PP1 inhibitor sequesters PP1 and makes sure that that is inactive. Again, we don't want to be having a kinase doing all this work to do a phosphorylation cascade, only to have a phosphatase undoing all that work. So that's the low blood sugar state. That's what happens when we've got to remedy a low blood sugar level by breaking down glycogen. Let's go over to the flip side here. Again, high levels of blood sugar means that your pancreas is secreting insulin. So again, insulin, fed state signal. We're going to have inhibition of adenylate cyclase because we're not activating it. So cyclic AMP levels are going to be low. So that means pKa is inhibited because those regulatory subunits are still bound to the catalytic subunit. So that whole phosphorylation cascade is quieted. So what that means is phosphorylase kinase without a phosphate is inactive. The serine 14 on phosphorylase is not phosphorylated, meaning that phosphorylase is inactive. Again, removing a phosphate or not having a phosphate on synthase means that we're synthesizing glycogen. All kind of consistent with what's happening here. But let's imagine we're just flipping over into this kind of fed state signal. What's really going to become important here is how these phosphates have gotten removed, and PP1 is again responsible for that. So again, if we've got insulin around again is going to activate, and this is the one thing that was a little bit weird, this insulin stimulated protein kinase, which is actually going to phosphorylate PP1. When PP1 is phosphorylated, it's actually going to be doing all of the dephosphorylation here. Because remember, we're toggling these guys back and forth. We're regulating these enzymes. This isn't a different enzyme that we've made. We're taking enzymes that were just maybe a while ago active to be breaking down glycogen, and now we have to make them inactive so that we can be doing our synthesis chemistry. So again, the important thing is that when we've got high blood sugar, that insulin signal is going to be what activates PP1. Now what's a little bit wonky is the fact that it's an insulin-stimulated protein kinase. So PP1 is actually active when we have one phosphate on it. We didn't get into the structural details of that. I kind of removed that from our learning unit this time around, but just understand that that's a little bit odd. But once PP1 is active, removing all of these phosphates, and importantly, when we don't have a phosphate on our PP1 inhibitor, that ensures that this guy's able to do its chemistry. All right, so lots of details there on the specific enzyme players in this cascade. The other thing that's really important is to think about how this is differential and differentially regulated depending on the tissue type that we have. So we've got muscle cells, and then we've got liver cells. Okay, so you have a few homework questions that allow you to kind of walk through this thought process. All the answers are on this slide, so I'm going to kind of walk through them one at a time here. So we know that our muscle cells and our liver cells both store glycogen, but they're able to help differently. So let's talk first about muscle cells. Muscle cells only can help and produce glycogen locally. And the main reason for that is twofold. They actually don't have the enzyme G6-Pase, and they only have GLUT4 receptors. So let's first talk about this G6-Pase activity. Just highlighting over here, if you break down internal stores of glycogen, it's made into G6P. If you want to export that, you have to have the phosphatase that will snip off that phosphate, make it into free glucose, and allow it to be exported. So not only do you not have the ability to make the free glucose to be exported, the receptor or the transporter that you have actually for transporting it wouldn't actually even be present there. GLUT4 receptors are only present on the surface of muscle cells when there's high blood sugar levels. When there's low blood sugar levels, because these are insulin-stimulated glucose receptors, they're actually not on the cell surface, so they won't even be there to export glucose. Let's flip over to the other side and talk about the liver. So one of the things as we kind of start beginning to think about things on a tissue level is remember that things like muscle cells we're going to call sort of selfish. They're selfish in the sense that they don't really take care of global needs. And you can think about your liver as sort of being one of these tissues that's sort of very motherly, sort of takes care of everybody else, makes sure if you've got garbage that needs to be cleaned up, it takes care of it. So the same is true with liver when we think about how it helps to remedy global blood sugar needs. So again, if you've got a global need for blood sugar, what's going to happen is your liver is going to break down its stores. It has G6-Pase, so it's going to break that glucose 6-phosphate down into free glucose. And the transporters that it actually has on its surface, and we'll talk about and dive a little bit deeper into those transporters in a minute, are able to export that blood sugar out. Just to kind of highlight here, both of these glucose transporters work in a facilitated diffusion manner, which means they are going to move glucose from high concentration to low. So the fact that these are always present on your liver surface means glucose is either going to be in or out of the cell, or moved in or out of the cell, depending on whether you're in a need state or a fed state. So again, that's one of the main differences between these two glycogen reservoirs, is whether they're able to help locally or globally. Okay, so how they're able to respond and help in those situations is dependent on sort of 4 signals here that we have that are represented by these different colored circles here. So the question on your homework asks you to think about what those signals are, how cells will respond, and whether one or both of these tissues respond. So let's first think about epinephrine. Again, epinephrine is released by your adrenal glands as kind of a fight or flight response hormone. So if we think about the logic here, it makes sense that both of these tissues would respond to it. If you're in a need where you need to either fight or flee, it makes sense that your liver is going to be breaking down glycogen and releasing it globally so that every cell in your body has glucose to do what it needs to do. Your red blood cells are going to have glucose to function and deliver oxygen. And importantly, your muscles want to respond to this signal as well, because they're going to mobilize their own glycogen reserves to glucose so that your muscles can do whatever you need to do to either fight or flee. If we think now again about another need state signal, but we think about your pancreas secreting glucagon, as we talked about above with kind of the local versus global, it makes sense that only your liver is going to be able to respond to glycogen, because really only your liver has the machinery to deal with exporting it. Muscle cells don't have G6-Pase to be able to even convert it into free glucose. And even if they did, if we've got low blood sugar levels, you're not going to have glucose transporters on your muscle cells. So even though you're breaking it down inside and you might have a higher concentration here than you do externally, there's no transporters there that would allow it to exit via facilitated diffusion. Again, contrast that with your liver. Your liver is going to respond to glucagon. It's going to break down glycogen stores into free glucose. And because these transporters are always present, high concentration of glucose on the inside as you're breaking down your glycogen will allow that to be exported to your bloodstream to help remedy that global need. Let's look on the flip side and think about more fed state signals. So again, glucose and insulin sort of kind of connected to one another, where if you have high blood sugar levels, you're going to have your pancreas secreting insulin. It makes sense that both of your cells will actually respond to this signal because they're both going to be able to help and pull in glucose. As we'll see in our next slide, they're actually going to help in a little bit of a different way, because when muscle cells pull in glucose, they pretty much just fill up their glycogen reserves, and then when they're done, they're done. They're not going to pull in anymore. Your liver cells will pull in glucose if it's high, but they'll first kind of fill up their glycogen reserves, and then once they're done, if there's still excess blood sugar, they're going to have to figure out how they can help deal with that. And so as we talked about before, you're actually going to continue to metabolize that glucose into acetyl-CoA, but then you're going to then store it as triglycerides. So one of the things that we'll delve a little bit more deeper in future chapters on is why there's different amounts of insulin receptors. There's greater insulin receptors on your muscle cells than there are on your liver. And part of this too is the fact that your muscle cells are going to kind of get first dibs on glucose that's in your bloodstream. And your insulin receptors on your liver are really only going to say, let's take in glucose only after everyone else has been satisfied. Let's take care of other cells first before we pull that glucose in for ourselves. So a couple of things just to highlight. Another homework question asked you to think about the differences between the kinds of epinephrine receptors that you might have on your cell surface. So even though your adrenal gland is secreting one molecule as epinephrine, there's different kinds of cell receptors on your surfaces that will respond to the same signal but have a different secondary messenger on the inside. All that I really need you to know right now is that beta-adrenergic receptors are present on both cell surfaces. They're the ones that stimulate adenylate cyclase, and so they're going to increase cyclic AMP levels and facilitate sort of this breakdown of glycogen. Alpha-adrenergic receptors, on the other hand, epinephrine binding to alpha-adrenergic receptors actually increase the influx of calcium into cells. And so again, that is also part of the degradation process when we consider phosphorylase kinase, but it is a receptor that's unique to liver cells. We'll delve a little bit more deeply in future chapters as to why that is, but for right now, all I really need you to know is that there's two different kinds of receptors. Beta-adrenergic receptors activate adenylate cyclase, and so they are going to increase cyclic AMP levels. Alpha-adrenergic receptors are responsible for the influx of calcium. So the last thing to think about before we kind of move on and pull this all together is the different kinds of glucose transporters that we have here. So GLUT4 transporters are present only in muscle. I shouldn't say only in muscle, but differentiating between muscle and liver here, GLUT4 transporters are insulin sensitive. So they're only going to be circulated to the cell surface when there's high levels of glucose. So again, that allows them to really be a one-way delivery system of glucose in when blood sugar levels are high. GLUT2 transporters, on the other hand, for liver, are always present. So because they're always present and they always operate in a facilitated diffusion manner, they're going to be moving glucose in under high blood sugar levels and allowing it to be exported out when we have low blood sugar levels and a global need for glucose. So we'll delve a little bit deeper in some future chapters about how this works and how we really want to kind of regulate the flow of glucose in and out of liver. It has to do with some enzymes that are inside, but we're going to table that for now. All right, the last thing to do to pull this all together is to think about this tissue regulation system and how that is different with regards to muscle versus liver. So let's start with our high blood sugar situation here. Okay, high blood sugar means glucagon is low, insulin is high. Again, insulin is going to stimulate GLUT4 to be mobilized to the surface of muscle cells. Insulin is also going to stimulate that protein kinase that's going to phosphorylate PP1 and basically make sure that we're put into a D-phospho system. High sugar, fed state, D-phospho system. So again, an insulin-stimulated protein kinase is going to be activating PP1 to do that. Again, now that we've got our D-phospho system activated, that's going to be synthase being activated, polymerizing glucose. We're going to fill up our glycogen stores in our muscles, and then basically that's going to be continuing to happen until those stores are full or until blood sugar levels drop to 5 millimolar or lower. In that case, your insulin levels are going to drop, your muscle cells will resorb those GLUT4 receptors, and they're not going to be pulling in glucose anymore. That's kind of an important protective measure, right? If your blood sugar levels have dropped, you need insulin to drop because you do not want these glucose transporters to continue to pull in glucose from your bloodstream, because they can actually cause hypoglycemia in that way. Let's contrast that with the liver. Everything's going to be the same except this first step here, because it's a different transporter. We've got glucose coming in via GLUT2. Again, GLUT4 transporters are actually going to be more aggressive in uptaking their glucose. They're going to get kind of first dibs at it. Anything that's left after the muscles have pulled in what they need will continue to go into the liver via GLUT2. Same processes happen here. Insulin-stimulated protein kinase is going to put us in a D-phospho system with PP1. Synthase being activated is going to synthesize and polymerize glucose in the liver cells, storing it as glycogen until those storage reserves are full. And at this point, we differentiate a little bit with what happens with muscles, because if there is still excess blood sugar in the bloodstream, liver will continue to pull it in. We will have glycolysis, and then pyruvate dehydrogenase making acetyl-CoA, allowing us to store those reserves as triglycerides. So again, flip side here, we think about what happens with low blood sugar. This quadrant is actually the easiest to think about, because in high glucagon, low insulin sort of situations, your muscles not having the ability to respond to glucagon basically have no response. So muscles don't even know what's going on necessarily with low blood sugar in terms of this hormonal cascade. However, when we think about what happens in the liver, high levels of glucagon is going to stimulate adenylate cyclase. We're going to activate the synthesis of cyclic AMP, increasing the activity of pKa. That whole cascade that we talked about in our first section here is going to activate phosphorylase to break down glycogen. Remembering that glycogen gets broken down in kind of this 90-10 mix of G6P and free glucose, that free glucose coming from the debranching enzyme, that hydrolysis activity of that debranching enzyme. But if we need to export this for global use, we're going to have a G6Pase, phosphorylate G6P, making it into free glucose. It will exit via facilitated diffusion via the GLUT2 transporter, and will help remedy that low blood sugar need. Kind of an extra piece to this that wasn't kind of part of this discussion that we had, we'll learn about it in our next lecture, is thinking about how gluconeogenesis from sort of the bottom-up approach can help to synthesize glucose. We'll talk about this specifically in our next learning unit, but just to highlight here, gluconeogenesis primarily happens in the liver. It does not happen in muscle cells, but this is, again, a process that primarily happens in the liver as this bottom-up approach to remedy low blood sugar levels.